Hello, I'm NAYARA NASCIMENTO, and I represent the importance of cell banks in biopharmaceutical production as my conclusion work for the biotechnology specialization. Those are the list of contents that I will present you. Cell banking definition, types of cell banks, master cell bank, working cell bank manufacturing, maintenance of cell banks, drug development and biopharmaceutical production, and last but not least, CGMP cell banking. Cell banking can be defined as a systematic process of preserving and storing cell lines under controlled conditions for future use in various biotechnological applications. The purpose of cell banking is to maintain a renewable and stable supply of well-characterized cells, ensuring cell viability and functionality over an extended period. Cell banks are categorized into different types based on their purpose and role in biotechnological research and manufacturing. The two primary types of cell banks in biotechnological manufacturing are the master cell bank and the working cell bank, each serving distinctive functions in the preservation and utilization of cell lines. The master cell bank is a critical starting point in the cell banking process. It comprises a large quantity of well-characterized and validated cells that serve as the primary source for generating working cell banks and subsequent production cell lines. The master cell bank is a core element in biotechnological development, ensuring that the same cell line is used consistently throughout a product's life cycle. The working cell bank is derived from the master cell bank and serves as the mediated source for production purposes. It is established to provide a sufficient quantity of cells needed for research, development, and manufacturing activities. Working cell banks offer the advantage of reducing the need for continued access to the master cell bank, thereby minimizing the risk of depleting the master cell line. Cell bank manufacturing is a crucial and systematic process that involves the expansion and cryopreservation of cells to establish reliable and reproducible cell banks. The process begins with the careful propagation of cells from the master cell bank or working cell bank. Under optimized cell culture conditions, cells are grown to generate a sufficient quantity for future use. This step is essential to maintain the genetic stability and functionality of the cells, ensuring consistency and reproducibility in downstream applications. Once the cells have been expanded, they undergo cryopreservation in individual vials or containers like small single-use bags. Cryoprotectants are added to protect the cells from damage during freezing and thawing processes. Sterility assurance plays a crucial role in maintaining the integrity of the cell lines. Adherence to aseptic techniques and clean room environments ensures that cell banks remain free from contaminants and microbial growth. Additionally, strict regulations such as current GMP regulations ensure that the cell banking process meets the required quality standards and safety protocols. The primary objective of cell bank maintenance is to ensure the long-term preservation of cell lines. This involves storing the cells in conditions that maintain their viability, genetic stability, and functional properties. Cell banks are typically kept at cryogenic temperatures, often using liquid nitrogen or specialized freezers, like plate freezers from single-use support. The cell bank activities require diligent monitoring and documentation. Monitoring of the health and viability of storage cells on a regular basis helps detect any changes or deviations appropriately. The cells should be routinely tested for cell viability, identity confirmation, and genetic stability. For example, regarding quality control tests, and check if they are properly performed to verify the absence of microbial contaminants. Cell banking forms the foundation for developing novel drugs and biopharmaceuticals. Researchers use well-characterized cell lines from banks to test the efficacy and safety of new treatments, accelerating the drug discovery process. As we can see here in this picture, regarding the biopharmaceutical production, we have the master cell bank, the working cell bank. From a cell culture, we can have the production of drug substance, drug product, then have the final product for patients. CGMP, Current Good Manufacturing Practices. It's a set of regulations originated and checked by the FDA, ensuring the quality, safety, and consistency of cell banking. By adhering to the CGMP guidelines, cell banks establish and maintain master cell banks and working cell banks with standardized protocols. CGMP is not only a regulatory requirement, but also means a confidence in researchers, biopharmaceutical companies, and regulatory bodies. Meeting CGMP standards allows cell banks to obtain necessary regulatory approvals, participate in clinical trials, and deliver biopharmaceutical final products, fulfilling critical quality attributes to the market, not only in the USA, but worldwide. And with this, we are at the end of this presentation. Thanks a lot.